Hi, it's Dr. Joe. And today I want to go over the morphological unknown, or the project number one. And so to do that, I'm going to kind of walk you through things. So in week 3 here, in addition to what we have in the lab, there is a separate area that says unknown project one, morphological unknown. Okay. So when you select that, it's going to take you to a lot of information. The first thing that you're going to look at is probably the data sheet that I've told you to to download before. So let's just take a look at this and then we'll come back to that. Okay, so on the data sheet, first of all, download it, save it, and fill it out. Now you can fill it out in 2 ways. You can type it directly onto the sheet, or you can print it, fill it out, but then you're going to have to type it in anyhow, because I don't accept handwritten work. I accept only typed answers. And so whatever word processor you have, you can use, but you will have to save it as a .doc or .docx or a PDF file in order to upload it. Okay, so looking at this, you'll see that there are 3 pages. And that the first page, it gives you directions, talks about binomial nomenclature. So that means that if you write something down, like the name of your species at some point, you need to use binomial nomenclature. Look over here and you will see that all the bacterial species that are presented here, and sometimes they're not spelled right, but I'm fixing that right now. And you'll notice, though, that all of these are in italics, and that the first letter of the genus is capitalized, but nothing else is capitalized. And that's what I expect you to do too. All right, so the difference between colony morphology and cell morphology. The colony morphology is what you're going to see on the streak plates, what you're going to see on agar, and each of those individual colonies was grown from a single cell reproducing itself over and over again as it's incubated. The cell morphology are simply individual cells from that that you can put on a slide, look at under a microscope, and do some tests on. Okay, so you're going to get images, and you're going to use the information in Blackboard and in some of the data files that I'm going to show you to figure things out. Okay, so the colonial morphology table, you don't have to do. I'm going to take that out for you. But the possible unknown species are here, okay? And the colony morphology. Right here is where you would enter the information about what your colony looks like. So you can go back to the colony morphology PowerPoint that we had and take a look at some of the colony morphology. Okay, so here you have Bacillus species, and you can see various things about it. And if you go back here, you can say shape. Okay, what are we talking about when we're talking about shape? We are talking about the overall shape of the colony. Sometimes you can see it, sometimes you can't. Okay, so overall shape of the colony looks kind of round here. Okay, so this is Staphylococcus aureus. This is going to be Proteus mirabilis. And so from these, you can compare what you have to these, and you can look at what your shape, your margin, your elevation, your texture, and your color is. Now, this will definitely tell you what the identification of your unknown, because several of these look similar to one another, and you're going to need more information to figure out what it is. However, you would then digitally insert a color photo of your streak plate here. And then the next thing you're going to go to here is going to be, how did yours stain? Okay. The color of your stained cells, are they gram-positive or gram-negative? And then their morphology and arrangement. So in order to review that, you can go over to the cellular morphology PowerPoint, and you can take a look at the gram stain. And you can see here with the gram stain that you have these gram positive cocci that are arranged in clusters, and you have these gram negative rods that seem to be in clusters also. So here you have the gram positive cocci in chains. Here you have them in clusters. And here you have something called a tetrad. Now this is a negative stain, but it is a gram positive organism, and they are in tetrads. They're in groups of 4. So here is example of the tetrad. Here are the gram negative rods. Here are gram negative rods in chains. And we are not going to have spirillum. We're just going to have rods, and we're just going to have rods and cocci, okay? So in your lab report here, not only does it ask you what yours are like and to put your picture, but you need to figure out possible unknown species for some of these. This is worth 8 points, and you need to figure out if this is gram positive or gram negative, what the morphology and arrangement is it catalase positive or catalase negative, and is it motile? And we're going to talk about motility and catalase tests in lab this week. So you're going to have that information. But where do you find this information about these? We didn't have those in slides. We didn't give all of that to you. But it is there if you will look in the, and now I'm really, if you go back to the project, and I'm just going to go back to the weekly schedule and go to week 3, and then go to that project again. And in the project, if you look down here, there are data tables to help you figure out the unknowns. And so there are lots of data tables that we have from the Berge manual. And since you don't have access to that easily, it is in the library. So you can see there are charts here with all sorts of information. Not just information for this project, but information for the 2 projects to come. All right. And when you look through here, you'll see, there's E. coli. That's one of them. If you go up here, you're looking for bacillus, and bacillus isn't here. Oh my goodness, what are you going to do for bacillus? Well, there happen to be other tables. Okay, so you have to go through these and you have to figure these out. You're going to have an opportunity to work on these, until they're due on the 17th. So there's a lot of information here. And you can use this for this project and for the other projects. Let me close it now. And go back to, wait a minute. I don't think you got to see that. I think I just had it. Yeah. So these are the flow charts that you will find under the project. And like I mentioned, you can use them for this and also for the other projects you're going to do. Push this down a little bit. Okay, so that will get you through these. If you have a GroupMe study group online, you can talk about where you found things, but you should be familiar with using these to get information on your own rather than just relying on copying it for people. Now, what if you can't find something here? As a last resort, you can go out online and look for things. You can look at the American Type Culture Collection, and you can look at microbiology sites. And actually, some of these might be in some of the videos you're going to look at, looking for cell morphology catalase and motility. Okay, and then there are differential stains, acid-fast or endospores. Now, you should remember that when I talked about endospores, I said there were only two genuses that would have endospores, Bacillus and Clostridium. Bacillus, that's the only one we have here. Acid-fast, does it have mycolic acid in its cell wall? Only one of those will have that. And we talked about that. So you would fill this out. Is it acid fast or endospores? And you should be able to figure this out from our lecture material. And then you would look at your sample. And did your sample have a positive acid fast, or did it not? Was it not there? Did it have a positive endospore slide, or was it not there? So if you need one of these, it says to email me with your unknown number, but the unknown number that I gave you is going to have that in it. But if you think that you still need it, and you're in trouble, go ahead and email me or text me and we'll figure it out. Okay. And so if the photo is of an acid-fast stain, then you would label that. If it is of an endospore stain, you would label that. If you don't have one of those, put NA on here. Don't leave it blank. And then the final determination of your unknown species, unfortunately, goes on page 4 because of the spacing, et cetera, et cetera. So you would put your unknown species name here. Remember to use binomial name nomenclature, capitalizing the first letter of the genus with everything else in lowercase, and then have the entire thing in italics. So that's how you would go through this, filling out the report. In order to find, okay, so I'm going to get rid of that. Okay, so that's the report here. In order to find your data folder, you would come up here. So I've emailed you with a number, and you'll see that there are unknown numbers here with data folders. So let's go to this one and see what we have. There's a streak plate. The gram stain's not coming up on this. This isn't one anybody has, but you would have a gram stain here. You would have a catalase test. You would have a motility test. And this particular one has an endospore test. Now, if you can't see it really well, you can open it and you can zoom in on it. And on this one, there is malachite green. There are endospores. So this is either going to be a bacillus or a clostridium. So you should be able to find your particular unknown here. So the identification sheet is here. The folders are here. And then these are the data tables to help figure it out in the Unknown Project 1 Morphological Unknown, which is linked in your week 3. This isn't due until week 4 on September 17th. So you have some time to do this, but I wanted to go through it and so that you can be more comfortable with it.